Hello and welcome everyone to this video podcast for Epigenetics. In this podcast, we're going to continue our review of some of the basic concepts from genetics. And here I want to just kind of review some of the concepts behind inheritance. And in that discussion, we'll review meiosis and mutations. As we're thinking about inheritance, I think it's good to take a historical perspective and think about when the first group of people started thinking about maybe studying inheritance. And we can trace that back to 10 to 12,000 years ago. It probably happened before then, but we have some evidence that it at least started here with the peoples of this time practicing domestication of livestock and plants. They may not have thought of themselves as geneticists, but they really were practicing genetics. Next, let's think about 400 to 300 BCE. And during this time, people started studying inheritance in a more defined way. And one of the early thoughts about how inheritance occurs came from some scientists that you probably have heard of, Hippocrates and Aristotle. They came up with this idea called pangenesis. And talk about pangenesis, let's think about a whole human being here. And you'll be happy to know that my drawing skills have not improved. But here is an oddly shaped human being. And pangenesis proposes that there are these little inheritable units all over our body that migrate to the gametes. And in doing so, they would provide instructions to the gametes on how to make the next progeny. We now know that pangenesis is not accurate. And there are some common observations that would tell us that it isn't accurate. For instance, if this person here had lost an arm, maybe they're really unfortunate and they also lost a leg, poor fella, and he had children, you would predict that his kids would also lack a leg and an arm. But that's not true. There's no reason to think that would happen at all. In fact, his children should have an arm and a leg because little genetic parts from all over the body do not migrate to the gametes to provide instructions. So this and other kinds of common observations really discredited some of the ideas of pangenesis. There was also some experiments done, and we talked about them in genetics. Another interesting idea about inheritance also came up around about the same time, and this was the idea of preformation. The preformation hypothesis stated that within either the egg or the sperm, there was a tiny individual. I can't draw it into sperm, I'll just draw it out here, but they would have believed this little tiny human was residing in the sperm. Those who believed that the little human was in the sperm, they were known as ovists, and they believed that once the sperm fertilized the egg, it somehow another energized this little human to start growing and developing. This other group down here, known as the spermists, believed that once this sperm entered the egg, that it would be released and be allowed to grow and develop within the egg. And we know that this is not supported because if the ovists were correct, then you would predict that the progeny would look predominantly like mom, and if the spermists were supported, then the progeny would look predominantly like dad. And we know that progeny typically look like both mom and dad. So again, this was out. Then another hypothesis grew that we call the germplasm theory. And the germplasm theory says you have your humans here, and all the genetic information necessary to be passed on to the next generation resides exclusively in the gametes. Mendel, through his experiments using the pea plant, was able to show that indeed the germplasm theory is supported. That gametes from one parent contain all the genetic information necessary, and gametes from another parent contain all the genetic information necessary that together will produce the next generation. And we know that the germplasm theory is our current understanding of inheritance, and it is the one that is most supported. Now, this being an epigenetics class, I think we should mention this other idea, that is the inheritance of acquired traits. And essentially what this says is that a person's life experiences Maybe they are an excellent athlete. Maybe they're an excellent musician or an excellent artist or a top scientist. Those traits that were acquired over their lifetime, according to the inheritance of acquired traits, would be passed on to their kids. Now, by and large, this idea is also not supported. However, the more we learn about epigenetics, the more we realize that the experiences of an individual can alter the expression of genes in an epigenetic manner in that person's gametes. And as a result, what that means is that a person's life experiences can have an effect on those gametes and therefore can have an effect on the next generation independent of that individual's DNA sequences or the mutations found in that person. Again, life experiences can affect the expression of genes in this individual's gametes, which is similar to the thinking of inheritance of acquired traits. Not exactly, which opens up the door that we might be able to pass on things that we learn or experience in our lifetime. What I'm going to talk about now is meiosis. Though don't worry, we're not going to go into the nitty-gritty details of meiosis, just kind of the big concept, big take-home points of meiosis. And I also want to talk about gametogenesis. Let's begin by talking about meiosis. And you no doubt have had meiosis beat into your brain for many years whenever you study genetics or biology in general. But I do want to remind you that in meiosis you start off with a diploid cell, and then this diploid cell, through 2 rounds of meiosis, generates 4 haploid cells. Now there's a little difference between egg and sperm here, but we'll talk about that in just a moment. But in general, what we're going to say is we generate 4 haploid cells. Then these 4 haploid cells go through a maturation process to make gametes, either eggs or sperm. And I also want to make sure it's clear that in making these 4 haploid cells, a great deal of diversity is generated. For this class, you don't need to worry about where it's generated or how it's generated, but these haploid cells that are produced generate that diversity. And that diversity is, by and large, up until this class, we've talked about the sequence of the genes. How does that change and how does that affect the gametes and then the next generation? In genetics, we didn't talk about how epigenetics can also affect the diversity of these haploid cells, but we will talk a little bit about that in epigenetics. Okay, so this is all you really need to know about meiosis, or to remember about meiosis for this class. Now I do want to talk a little bit about this maturation process, but not in great detail. We'll talk a little bit more about this as the semester goes. But for now, what I'd like you to know is that the specific process of making gametes in sperm production is called spermatogenesis. And the equivalent process in producing ova or eggs is called oogenesis. During spermatogenesis, the process starts with a spermatogonium. That is the diploid cell, and it goes through meiosis to produce those 4 haploid cells that we'll call spermatocytes. And then those 4 spermatocytes will mature into 4 sperm. Now this whole process in males doesn't start until the male enters puberty. Oogenesis is a little different. It also starts off with a diploid cell, which we'll call the oogonium. It will go through meiosis to generate 1 unequally sized oocyte, because even though the nucleus divides and is segregated into 4 cells, there's not an equal division of the cytoplasm. More of the cytoplasm goes into one egg. So these are oocytes. These oocytes then will mature, or rather I should say that this oocyte will then develop into the mature ova. Now, as I mentioned, in males, spermatogenesis doesn't start until puberty. However, oogenesis starts much earlier. It starts when she is a fetus. And as a fetus, meiosis will begin, but it will pause during fetal development and will not pick up again until puberty. It's possible that this very long pause in oogenesis may account for reprogramming of the expression of genes in the oocytes due to different things that she might experience, which would be a link to epigenetics. In fact, during both processes, there's evidence that what the male or the female experiences does affect the gene expression during spermatogenesis or during oogenesis, which is then passed on to the children. The last thing I want to talk about is the difference between somatic and germline mutation. We spend a lot of time talking about mutations in genetics, and we don't really want to focus too much on that, because epigenetics, again, as I've said once or twice or maybe 1,000 times, doesn't have anything to do with mutations found in the gene that's being regulated. But I do want to introduce or reintroduce these two terms to you: somatic and germline mutations. Somatic mutations occur in any cell except germline cells. That is, except the sex cells. Somatic mutations are not inherited. When these mutations occur, they affect a localized region. If I get a mutation in my pancreas, it's going to affect only those cells found in my pancreas, and maybe only a very small region in my pancreas. It's going to have no effect at all on my liver or any other part of my body, just where the mutation occurs. Germline mutations, however, these only occur in germline cells. Those cells destined to be sperm or egg. These are inherited. So that's a very important point here, because if a mutation occurs in a germline cell, that means in the next progeny, every single cell of that individual will have that mutation. So I'll write that here. Mutations appear in every cell of the next progeny. I bring this distinction up as we begin our discussions with epigenetics, because epigenetic changes can occur in somatic cells, and they can also occur in germline cells. And they will have those same kinds of features. When they occur in somatic cells, they will not be inherited, and they will only affect that localized region. Same thing with epigenetic changes in germline tissues. Those will most often be inherited to the next generation, and that will affect that next generation. Okay, that's all I have for this podcast. If you have any questions at all, please make sure you come talk to me. If not, I'll see you in class. Bye for now.